Sure, yeah. And just as a reminder, sorry, is this? Oh, it doesn't. This is probably not. Oh, it is working. Sorry, yeah. As a reminder, the target for therapeutic oligonucleotides is almost always RNA, and usually messenger RNA. And the great thing about oligonucleotides is that they can arrest the synthesis of proteins at the mRNA level in a way that most small molecule drugs, or pretty much all small molecule drugs, can't really do. So this enables us to address diseases and different types of conditions which are not addressable using conventional small molecule therapies. And that's a huge amount of the genome that we can then cover with oligonucleotides. And the design is obviously really simple because we just need to know the sequence of the target. And we always know that these days because of DNA sequencing. Now, this is just a review of the oligonucleotides which are currently in the clinic. And I won't dwell on this because the key point here is that there are three types. There is antisense oligonucleotides, which depend on RNase H to degrade the RNA. Exon skipping oligonucleotides, which actually make mRNA make proteins that they're failing to make. And siRNA, which is double-stranded, relies on the RISC complex, and destroys messenger RNA. So we have three different modalities, and I've color-coded them. And as you can see at a glance, it's a pretty even split between the three different modalities. There are other modalities which are coming along. There's microRNA and a number of other modalities. But the key point is that in recent years, the number of oligonucleotides coming into the clinic has really expanded. The field is very active. Now, we've done a lot of work recently on the exon skipping applications, whereby we take a pre-messenger RNA, because mostly pre-messenger RNAs are actually spliced to make messenger RNA, which makes a protein. But in certain genetic diseases, and the classic example is Duchenne muscular dystrophy, the disease is caused by a mutation. And the mutation either puts a stop codon into the mRNA, or it throws it into the wrong reading frame. Therefore, the protein that's synthesized is not functional. Now, it's quite remarkable that what you can do is, with one of these mutant mRNAs, you can block the site of the mutation with an oligonucleotide when it's carefully designed, and that oligonucleotide can put the reading frame back in place. Now, it does also leave a small patch of the protein that is not made. So it's a truncated protein, but can be partly functional. And that's the kind of example for a good example of that is Duchenne muscular dystrophy. And there are several oligonucleotides in the clinic for different mutations of DMD. But the problem is that with therapeutic oligonucleotides is that a standard unmodified oligonucleotide clearly is not going to work, because cells have the ability to degrade oligonucleotides using nucleases, and the lifetime of oligonucleotides in cells is incredibly short. So we have to develop chemically modified oligonucleotides, which have got good stability, good RNA affinity, good specificity. Low toxicity is obviously crucial. They have to get into cells, but that's not the whole story. Even when oligonucleotides get into cells, they tend to reside in endosomes and are either degraded or very slowly released. So endosomal escape is important. And what's becoming more and more important as the field develops is to be able to target specific organs or cell types. And this is one of the key areas of at the moment of development. So these are quite a high bar to get an oligonucleotide to work in cells. So we can't use unmodified oligonucleotides. So in order to make oligonucleotides stable in cells, what we tend to do is put a phosphorothioate group in them, which the sulfur gives some kind of resistance to degradation. There are two isomers, of course, it's chiral. We can also add sugar modifications, which further stabilize against degradation in cells. And we can increase the target specificity by using modified sugars like LNA or constrained ethyl, which are just two related sugar modifications. All these kinds of modifications can be used in exon skipping oligonucleotides, but the modification that's been most popular and important is this PMO exon skipping backbone, morpholino backbone, which is charge neutral and is very different from a normal phosphate group. The classic exon skipping oligos in the clinic are based on this kind of modification, which is charge neutral. All the other modifications here have a negative charge. So charge neutral backbones have become important. PMOs are a good example. And here are the 4 oligonucleotides which are used to treat different mutations of DMD, which are based on PMO technology. So we wanted to know whether or not we could do something along these lines, but in a much simpler way, a more flexible way, by working on charge neutral backbones in oligonucleotides. And that will be relevant to all 3 modalities of oligonucleotide therapeutics, which I can discuss offline. So we went into this area and we made a whole number of different triazole linkages and a number of linkages which I'm showing on this slide. This is the conventional phosphorothioate with a sugar modification. We made amide linkages with LNA, carbamate linkages, extra carbon there on the carbamate, and sulfamate linkages. And we studied these modifications and we published this work quite recently. And, the different modifications have got different kind of biological properties, advantages and disadvantages. But this particular one here, I think Afaf might be talking about this later, is particularly interesting, the sulfamate linkage. But we wanted a very simple way to make oligonucleotides that contain charge neutral backbones that anybody can do on a DNA synthesizer. So our idea is as follows. We take a conventional nucleoside, a DMT protected nucleoside. This one's LNA. These are made in huge amounts for therapeutic oligonucleotides, which are currently in the clinic. So this particular molecule, you can buy this. It's the final stage intermediate for, the majority. It can be LNA or methoxyethyl or O-methyl. It can be different sugars, but you can buy all these and they're readily available. And we phosphorylate that particular alcohol with a specific phosphorylating reagent where we have an R group here. And this R group is what gives us the charge neutral linkage. Normally, this would be a cyanoethyl group, but we don't do that. We use a different phosphorylating agent. And we end up with a set of monomers that we can use directly and routinely in oligonucleotide synthesis. And we can combine this R group. It will normally be actually an alcohol, as I've shown here. It can be an alkyl group, but more commonly we use an alcohol. And we can put a sulfur here. It can be a phosphorothioate or it can be a phosphate. It doesn't need to be a phosphorothioate in terms of stabilizing the oligo against degradation, because the triester that we're making here, this triester, is stable to enzymatic degradation anyway. But we can include the sulfur if we want to include the sulfur, because sulfur also increases cell uptake. So we can do sulfur or oxygen here. And what we found very briefly, and it's all published, is that if this alcohol is a primary alcohol, the triester is fairly unstable, chemically unstable, particularly if you heat it in ammonia. If it's a tertiary alcohol here, the tertiary alcohol comes off and you end up with a conventional oligonucleotide with a phosphodiester backbone. But what we found and what people have kind of missed in the literature, if this is a secondary alcohol, we have a stable linkage and it's easy to make and easy to work with. So almost every modification I'm going to talk about has a secondary alcohol attached to the triester. So this work has been done by Debashis, who was a postdoc in my group, who's done a huge amount of work in this area in the last 2 years. Remarkable amount of effort that he's put into this. And as I mentioned before, we can vary the backbone, sulfur or oxygen. We can vary the sugar, LNA, OME, MOE, and F. And we've done all those. We've made all those as triesters. And we can vary the R group. And the point here is we can vary the R group so much. We can put so many different alcohols in this position in the phosphate backbone to give a charge neutral triester backbone. I've given a few examples, simple alcohols like isopropyl, longer alcohols, cyclic versions, alkynes, which I'll come on to for obvious reasons, fluorocarbons, lipids, glycols, all sorts of things we can put into the oligonucleotide in a very straightforward way. And basically, if we use the alkyne linker here, if we make an alkyne triester, we of course have the ability to put anything we like onto that linkage, more or less, using click chemistry. So we just take this oligonucleotide, we synthesize the oligonucleotide with the alkyne in the backbone. Now, these oligonucleotides can have any number of triesters in the backbone. They can have 1, 2, 3, 4, or they can have a full triester backbone. But what we find, and I'll come on to that later, is that if we have a small number of triesters, we get advantages in biological media. But to give an example of the click chemistry approach, we've shown that we can put sugars, glucose, galactose, lactose into the backbone. We can put amino acids, lysine, leucine, phenylalanine, valine, and we can put polyamines like spermine. There should be one more nitrogen somewhere up here, spermine, to increase binding to target RNA. So that gives us a huge amount of variety. So we can make oligonucleotides with small numbers, large numbers of modifications within the backbone using this methodology. Now, this slide is just to show that we've made a large number of oligonucleotides. We've made about 200 oligonucleotides using this triester chemistry, but I've highlighted a few. The yellow ones here show that if we make an oligonucleotide and include 4 modifications, and if that modification is isopropyl, it's much more stable duplex against RNA, plus 13 degrees, than the conventional 2'-O-methyl phosphorothioate. So the LNA moiety that we attach to these bases increases stability of the duplex as it does with normal LNA. So we get a triester which has got very good affinity for its RNA target. If you look at the conventional LNA control, now it says not done here. What that means is that that is so stable that we couldn't measure the TM of that sequence in this way. days with PhD. But since then, we started working with ionic traits. So not only are we reducing the negative charge, but actually in positive charges into the alkaline. When we use this nitrogen instead of oxygen, we're increasing this activity by a lot more. So we're getting very good insights. Now, I want to summarize this piece of work and then published papers and 12 patents. Key points are that the synthesis of the oligos is pretty straightforward, and the monomers are easy to get, and the modifications. I can also say briefly, just at the end, it's also consistent with ASOs and SRI.
I wanted to thank Tom for giving me this opportunity to speak here. And I'll start off by jumping back to the slide with the central dogma to highlight the central role of the mRNA in gene expression. We all know mRNA from COVID vaccines, but because of the ability to encode virtually any sequence of protein of interest, we're able to go much further beyond just the vaccination, with examples as protein replacement, gene editing, or cancer immunotherapy currently in the clinic. Most of the applications that are now in the clinic rely on so-called cap-dependent translation, for which the central role plays a distinct modification at the 5' end of the mRNA called cap that attracts ribosomes through the interaction with EAF4F complex. This mechanism works really well in normal conditions, but as the field moves towards some more specialized applications that require targeting specific cell types or very efficient translation in specific conditions, like, for example, under cellular stress, this specific mechanism of translation doesn't offer much flexibility for optimizing those parameters, like, for example, cell types specificity. In contrast, we have also a natural mechanism of translation that's called cap-independent translation, and that relies on a secondary structure of mRNA called internal ribosome entry site, or IRES for short. And in contrast to cap-dependent translation, this offers some more space for optimizing such parameters as, for example, cell type selectivity, and also it offers reduced sensitivity to stress conditions. However, in general, translation of the cap-independent mRNAs is not as efficient as cap-dependent translation. And on top of this problem, we also have a lot of issues with the current strategy for characterization of the elements promoting cap-independent translation. The current approach relies on dual reporter by isostronic strategy that in fact uses cap-dependent translation for normalization of the cap-independent translation data to the uptake of the mRNA. However, because we have this component that's translated cap-dependently, it can interfere with the translation of the second C-strand because of, for example, reinitiation of ribosomes from the first C-strand on the second C-strand. It gives unreliable cross-cell type normalization because the cellular proteomes vary for different cell types. So the translation of the first C-strand can also vary. Of course, despite we are getting a lot of fluorescent molecules, they're not attached to the mRNA, so we cannot directly track the mRNA fate. So to solve the problems related to the cap-independently translated mRNAs, we question how we can actually turn the disadvantage of this linear cap-independently translated mRNA, which is lack of the cap, into an advantage. So you can see that this mRNA doesn't rely on the cap, so we can use this space to introduce some other chemical modifications.
And to do so, we used this dinucleotide in vitro transcription primer that's modified with azide at the 5' end. And you can use it by just adding it to a standard in vitro transcription reaction mixture. And by using T7-RNA polymerase, you can very easily incorporate it into the mRNA. And as you can see here, because of this dinucleotide design of this molecule, when we use at least 4 millimolar concentration of it, we can have more than 95% incorporation into the mRNA. And obviously, because we have azide installed on the mRNA, we can use it for further conjugation. And a good example is strain-promoted azide alkyne cycloaddition. So we can use a broad range of commercially available or easy to prepare strained alkynes to very efficiently conjugate them with the mRNA. And after around 30 to 60 minutes, we can have virtually full conversion. So we have the modified mRNA. In this, let's say, preliminary studies, we use a model molecule that was fluorescent dye, which is obviously fluorescent, but also serves as a bulky blocker at the 5' end of the mRNA, shielding it from exoribonucleolytic decay. For this modified mRNA with the dye, we observed around 8-fold improvement in the translation efficiency compared to unmodified 5' triphosphate, and similar improvement over the much smaller azide installed at the 5' end. This is partly due to the stabilization of the mRNA, but also due to the fact that we get rid of the problem of the immunogenicity of the 5' triphosphate, which is common in viral mRNAs. You can see here that for the 5' triphosphate version of the mRNA, we induce very strong expression of viral infection-related cytokines. But if we replace it with modification like azide or the fluorescent dye, we get rid of this expression of immunogenic of this immunogenicity. So I mentioned the problem related to the characterization of the elements promoting CAP-independent translation. And because we have the fluorophore installed on the mRNA, we can actually also use it to normalize the expression data to the cellular uptake of the mRNA. And because we are using fluorophore attached to the mRNA, it's independent on cellular factors, for example, related to translation. We can very conveniently use flow cytometry to assess the fluorescence of those two reporters at a single cell level, and we can compare different elements promoting CAP-independent translation. For example, in this study, we compared 3 different designs of the mRNA incorporating 3 different viral irises, and we compared how they perform in 4 different cell lines. So just to briefly walk you through the idea of the normalization, if you look at the sole protein expression, we can see that all of the mRNA designs were most efficiently translated in HeLa cells. And this is not surprising because HeLa cells are actually the largest out of all of the cell lines that we tested. But if we look at the uptake of the mRNA, it's also the highest for HeLa cells in all the cases. So if we combine this information and calculate the actual mRNA productivity, we can have much better image of the performance of the translation machinery in cells and how it reacts to different mRNA designs. From this benchmark, you could see that one of the constructs we tested called ISIN, or synthetic iris, performed really well. This design was originally optimized for circular RNA and reported in this Nature Biotechnology paper in 2023, where authors mentioned that in its linear form, it was performing very poorly. So we were actually very surprised to see how well it performs if it's just modified at the 5' end. And you can see compared to the other irises, very common actually in the field, which is from HCV virus, we get 67-fold improvement in translation. But if we compare this linear protected CAP-independently translated mRNA to the circular version as well as capped linear version, we can see that its performance is really close. And this is despite the fact that its 3' end is still unprotected, so it's susceptible to exoribonucleolytic decay.
And finally, I wanted to just briefly mention that you can use exactly the same constructs of mRNAs with the dyes attached to the 5' end to characterize the mRNA uptake and get some more mechanistic details on that. You can use flow cytometry as well as microscopy to track the mRNA uptake and protein expression to, for example, calculate what's the lag between the delivery and expression. You can also study the uptake of the mRNA in more detail. For example, here we studied a single transfection event where we could observe very nice diffusion of the mRNA inside of the cell and its entrapment in specific cellular compartments. Despite having only one fluorophore attached to one molecule of mRNA, we got really good signal to noise. You can track it further, for example, going through the process of cell division and looking how the mRNA behaves there. And obviously, you can use some techniques like, for example, spinning disk confocal microscopy to get three-dimensional insights into this. And you can go one step further and use fluorescent staining of some specific cellular compartments to know, for example, what fraction of the mRNA localizes in endosomes or lysosomes, which can be helpful if you're optimizing delivery strategy. So to sum up, we developed a strategy for improvement of the properties of cap-independently translated mRNAs, not by just directly improving their translational properties, but also by providing a toolkit for their characterization and tracking their cellular fate. You can find more details about this work in our recently published paper that's cited here. And finally, I wanted to thank my supervisor, Tom Brown, and my colleagues involved in this work, and Nature ETN Consortium for the funding. Thank you.